Alright, hey everybody, we're live. I'm going to give it a little bit to let people jump in.
Are you started?
Yeah. Just going to try to...
Wait, are you on?
Yeah.
What meds are you talking about, Andy?
Just some research on some meds, like the hydroxychloroquine. And then I have one pulled up. It's an antiretroviral.
What's it called? I wish I would have known. I had 2 meds in my lecture, and like they're.
I have it pulled up. I am not really prepared.
This is what I'll do. Because it's from my pharm class. So the mechanism, can you hear me?
Mm-hmm.
The mechanisms of action are like super in depth, down to the cellular level. I'll write it on a piece of paper, and if you want to mention it, you can.
Yeah. Cool.
Ellie.
Covering the miles.
Okay. All right. Let's get to it. All right, everybody. Just doing a little live video here. I know it's a little disorganized, trying to get hooked up into a group that I'm in, trying to see if they will stream it and approve it. So I had to make the title a little different. Couldn't go with the COVID. Facebook might shut that down. But basically wanted to just do a little video about what's going on. This is not a diagnostic video. I am not an MD. I am not here to tell you to go to the doctor, to see a doctor, to do anything of that nature. Just a little what it is. What is COVID-19? Also known as SARS-CoV-2. It's related to the SARS coronavirus, the virus that caused severe acute respiratory syndrome that was around in 2004, that also came from China, cross-species type of virus. So basically, if you need to see a doctor, go see a doctor. I think the guidelines are that you should call ahead if you are experiencing any of the symptoms. I think that'll be a good place to start in terms of what the symptoms actually are. It's fever, tiredness, dry cough. Some patients have aches, pains, nasal congestion, runny nose, sore throat, and diarrhea. I think diarrhea is under 10%. Aches and pains can be a little bit above, like 16%.
Also smell and taste.
Oh yeah, and then the main thing that everybody tends to say is loss of smell, like the sense of smell, and taste. So if you're having any of these symptoms, please call a medical professional, MD, DO, talk to a nurse, call your family physician, call ahead to any emergency room or urgent care that you might be going to. They will give you guidance as to how to proceed. So basically, let's, if you have any questions, just throw them into the chat. I'll be able to answer them. But I did want to touch on one of the myths that are going around, that this came from bats, that somebody ate a bat and actually caused the virus to jump. Yeah, I know, don't touch my face. I'm at home, I'll wash my hands. But it didn't actually jump directly from bats to humans. The working theory now is that there was a change in mammalian vector. Basically, it went from bats to one of the animals that are associated being around bats, a palanquin. I don't know if anybody's familiar with those. Those are really cute looking animals, but it stems from Eastern medicine and Asian cultures, particularly Southeast Asian cultures, defining them as a delicacy. And they transferred from bats over to palanquins and then palanquins to humans. That's the running theory as of right now. That basically comes from not having necessarily the same regulations that we have here. Yeah, I'll talk about Dr. Fauci in a second. Yeah, so it's not necessarily someone who went to a market and bought a bat, threw it in some soup, and did eat it. It has a long-standing cultural legacy that people will eat particular animals over in China or in Eastern cultures for medicinal properties. In particular, palanquin is associated with kidney or adrenal health. Obviously, that's unfounded. There's no clinical trials. It's whatever science you want to, whatever theory of science that is. Is Dr. Fauci making as seriously as is making it? Yeah, I think he's a bit of a tough position. The guy's been in there since Ronald Reagan. Been the head of the National Institute of Allergies and Infectious Disease since 1984. He's very well educated. The man is the leader in his field. He's in a tough position because he's in an apolitical position, meaning that he's not necessarily associated with Republican, Democrat, whatever. He was instituted based upon of his educational background, his achievements in his field. He's not there because he was appointed, like as say the Secretary of Defense. He was appointed there because he earned his degree. He did science. I'm not familiar with what his particular subfield is in infectious disease, but I know that his main research has been infectious disease. So I trust what he's saying. Yeah, 200,000 deaths are predicted. That's basically a couple of things. It's an updated number based upon models from the Italian outbreak, the German outbreak, the United Kingdom, South Korea, and China. Now, it may be a little biased of me, but I don't necessarily believe the numbers coming out of China, and that's because they have more of a secretive type of government. They have a little bit more control over the information that is allowed to leave. But if you look at Italy's model and you look at how contagious and infectious that this disease is, it looks to be anywhere between 2 and 2.5 as a score of an R0. R0 is just basically is a fancy word or a fancy designation that signifies how many people will get infected from one person. So an infection rate of 2.2 is the latest number that I've seen, or anywhere between 2.2 and 2.5, means that for every one person that's infected, 2.2 to 2.5 people will get infected from that. It's not as bad as, say, SARS or MERS, also closely related viruses in the Coronaviridae family. However, it has much less of a lethality than those two diseases. But the longer incubation period for this disease as an asymptomatic carrier, we'll get into a little bit of that detail in a second, what's the best way to phrase that? It kind of makes this more of a virulent or more of an aggressive virus in the fact that you can spread it without actually knowing. So when SARS outbreak happened in 2004, you had to be symptomatic to actually share the virus with other people. So you had to have a cough, you had to have that dry hacking cough, you had to have pneumonia to actually share your disease with other people. With this, the lag time between actually being virulent and showing symptoms, or being able to spread the disease and showing symptoms, is anywhere between 1 and 2 weeks, I believe, about 10 days. So first you usually don't have any symptoms. You can spread it. You can actually be what's called shedding virus. is that you're producing virion particles in your respiratory tract. It tends to start as an upper respiratory infection and then travel down into your lower respiratory tract. So you'll start to have it in your nose and then travel down through your trachea into your lungs. And that's when you develop the pneumonia-like symptoms. But let me circle back to the 200,000 deaths. That's basically because we're not going to see as much of an infection rate moving forward because we're doing the social distancing. A lot of people are saying that it's crazy, that we're crushing the economy. There's a lot of conspiracy theories going around. I'm not going to really touch into all that. I believe that we're really experiencing what we're experiencing because whatever happened in China, whatever the regulations on that open air market allowed some type of species barrier hopping virus to hop from whatever species of animal that would carry the coronavirus over into our species. That then led to human-to-human spread, and then that's where we're at now. I don't think you should put any stock into this thing that somebody made this virus and spread it across the world. There are some people that did, yeah, I know, right? My cousin's in the background calling bullshit. It's basically because if you look at the way the virus is, the genome, you would be able to tell if it was genetically manipulated. But with all the data that's out there, there's a couple papers that I can pull up and I can share on the comment section after this that has actually done full genome-wide sequencing of this, and it's not man-made, it's not man-manipulated. You'd be able to tell. There'd be some type of markers, basically primers. You have little inserts that you would put onto a gene that would make something more virulent, or ability to touch my face again. You'd be able to see that, because then when you sequence it again, say that this portion of the pen here is the primer that you'd use to insert into the genome, that marker would be there forever. You can't get rid of that. So you'd be able to backtrack that, and there's too many people working on it for that conspiracy theory to really hold water, in my opinion. But that's just my humble opinion on that matter. So basically, what this genome, I guess that's a good segue, what this genome, will it end up being a seasonal virus? Very good question. Speaking of the genome, the mutational burden and the mutational rate for the virus is significantly low. So if the virus runs its course and goes into enough people, asymptomatic carriers, low symptomatic carriers, and even higher degrees in that 20% range that actually need to see a doctor, it should build up enough herd immunity that it won't become a seasonal virus like the flu. The flu is a type of virus that can mutate very rapidly and year to year, so that the little prongs on the end of the virus that come off like this and actually attach to your cell that allows it to infect said cell changes very quickly. It can change at least 2 to 3 times a year. So that's why every year they have a flu vaccine that's predicated upon a lot of epidemiological research, some modeling to show which of those spikes is going to be the primary spike that we're going to have to deal with later on in the year. With this virus, though, the changes in the virion particles, what's called the S1, S2, and the R proteins, do not change that rapidly. Like I said, there's only been, between Wuhan and Italy, about 4 or 5 detectable mutations. 8 different strains. There could be 8 different strains. I'm not familiar with that. Coronaviridae is a family of viruses that actually encompasses many different coronaviruses. It could actually be that there's MERS, SARS, SARS-CoV-2. There's a murine version of this, which is a mouse version of this. There's several bat versions of this. They all are Coronaviridae, but they're not necessarily derivations of COVID-19 or CoV-2, SARS-CoV-2 or SARS-CoV-1. I don't think that they're necessarily relate. They're relate, but they're not necessarily what you would colloquially call different strains. Meaning that you're not going to have SARS-CoV-2 sub 1A, sub B, sub 2A, et cetera, et cetera. Now, there was some talk and some research that showed that there was a more virulent version of SARS-CoV-2 in China. However, it was more temperature sensitive, is what the data indicated, and so that the main virus that you're seeing is the COVID-2 strain that we're actually seeing, COVID-19. Now, that's very disputed data. That can also lead to some speculation as to why there might be multiple strains out there in news or out there in the zeitgeist, so to speak. Did you see the news article where they fired a nurse reporting the numbers that numbers are actually supposed? If you could actually show me that news article, I can definitely take a look at it. But I don't think there's an underreporting of COVID-19 in the United States. I think we're actually doing the best in the world in terms of reporting it, not necessarily in terms of our testing capabilities and the ability to track the virus from person to person in terms of spread. We have a slow start to getting testing out and in the open in comparison to other countries such as China, but in particular South Korea. South Korea did an excellent job containing the spread. They actually did a lot of tracing. They well-funded their version of the CDC that actually led to their ability to get on top of this and to basically squash it before it actually spread to the mass of the population. Yeah, I'm trying to avoid the political aspects of this. I will give credit where credit was due. I think the shutdown of travel from China was very intelligent. I thought, it may have been done for the incorrect reasons. However, the time that gave us to actually get on top of this was not spent well. I think that we could have increased testing capabilities in this country much more rapidly. We were still using and still are using what's called a PCR analysis, polymerase chain reaction analysis, to determine whether or not you are COVID-19 positive. They do now have in the works, I believe it might actually be on the shelves now. They were supposed to have it on the shelves or available at March 30th. There's an antibody test and a quick bedside reactivity test so that you can see whether or not you're reactive to the virus. So you take a sample, if it's reactive to the virus, boom, it's instant. What everyone was using previously was PCR analysis. So you take a little swab. I think everybody's seen the memes. I don't know if that's accurate or not. They go into the back of your throat, they take this collection, and then they get rid of all of the human cells, so to speak, get rid of all your DNA from that, and get viral RNA. They take the RNA, which is a molecule that expresses genetic information that is similar to DNA. We're not going to get into the differences right now, but you take that RNA, you make a DNA plate, and then you expand upon that DNA template to see if you're test positive for COVID-19. That takes a little bit of processing. You take the sample, process the sample, and then run the test and actually see if it matches up in terms of the length of the RNA, and if it matches the length of what's the markers that are known for COVID-19. I believe it's about 3 or 4 genes that they test for, and then that's your determination. That's your first primary test, and then they do a secondary test to confirm that, because there are some false positives that can come from that and false negatives that can come from that. It's not necessarily the most accurate of tests. It's not like an antibody where you can just have an antibody that detects the virus, boom, you get a readout right then and there. So basically, I think that covers a lot of it. So treatments right now are basically you use some soup, some yes, Remdesivir. Yes, I actually know that. So what do you know what they use it for in terms of, is it like HIV drug?
For Remdesivir, I think it's been used in HIV, but it was presented as a medication to previous corona strains, and it kind of went away because they didn't need it, and now they're reintroducing it.
So there's a couple things. I know that they're using.
It's an HIV drug.
I think it's an
I'm
HIV
pretty
drug.
positive it
So
does.
Remdesivir is it blocks RNA polymerase. So if we look at the virus itself, the coronavirus is a coronavirus is an RNA based virus. So basically, it's a single strand positive RNA virus. So that basically means that it's got a prime to 3' RNA as its main genetic carrier of information. Let's go back to that pen example. You have the spike attached to your epithelial through the angiotensin AC1, I believe, receptor. At that point in time, the envelope will go into the cell. The virus will be enveloped by the cell. Just looking at some of the comments. It's uptaken or absorbed by the cell, and then it is automatically, does not need to go through any type of modification. It doesn't have to go from DNA. It does not have to be inserted into the cellular genome. It can automatically be used by the ribosomes to produce proteins. There's 5 main proteins. There's the capsid. There are the spike proteins, which I was talking about, which actually allow entry into the cell. There are ribonuclease proteins. There's a helicase and a replicase protein. Those proteins are all used to make eight different strains. Look at the 8 different viruses, the strains. I don't necessarily think that they're going to be strains as in there's different mutations of the primary sample of the virus, but yeah, so even if there are 8 strains, they're so closely related to one another that these different proteins that are inside and outside of the genetic information of the virus, you're going to be able to develop a vaccine that will be able to target all of them. Again, there's some studies right now with the flu virus that they're actually not making it to these spikes anymore, like the H1N1, H6N6. Those are all different protein names for different proteins on the cell surface or the viral particle surface. However, they're looking at more of the body of the virus. So this is the body of the virus attached to there, and eliminate the virus that way instead of looking at it like this, attaching to this, because this portion of virus will change year to year, month to month. But back to some of the different treatments. I know that President Trump had indicated that hydroxychloroquine and its more toxic counterpart, chloroquine, could be used as a deterrent or could be used as a treatment for the drug. That comes from in vitro studies where about 2 micromolar of the compound, which is a very high concentration of the compound, can prevent viral entry into the cell. Again, that spike attaches to the cell receptor on the surface and then allows absorption into the cell. So I don't necessarily know if that's going to actually be used in vivo for, say, animal experiments, or in vivo or in human clinical trials. I believe some places are probably studying it. However, hydroxychloroquine tends to, I think, is in a class of drugs, antibiotics. It's primarily used to cure yourself from bacterial infections. So I highly doubt there's going to be that much crossover between the ability for it to treat COVID-19. That goes without saying. that I'm not a clinician, I'm not familiar with the clinical trial data, and it's still too early to actually know whether or not it's an effective treatment. Do you believe there is validity to those people using? Okay, both questions. NSAIDs. When you're seeing some complications with people that are getting NSAIDs, like there were some reports in France, that could be correlative rather than causative. And that's why a lot of people had tended to say and downplay the results of those studies. You could have had just some people getting ibuprofen or acetaminophen. Acetaminophen's not technically an NSAID, is it?
Tylenol's not.
Tylenol's not an NSAID.
Aleve.
But Aleve, naproxen sodium, stuff like that. That could have just been something that occurred concurrently with downward spirals of said people. I don't necessarily speak to the efficacy of NSAIDs. However, if your doctor recommends that you take NSAIDs, then you should follow your doctors. But there is no empirical data that I could see other than what's called anecdotal evidence to support those claims that NSAIDs are going to actually, yes, Advil, ibuprofen, Motrin, as brand versions.
Symptoms, they're not going to cure.
Who do you read? Greater than 95 use. Where do the NSAIDs? There was some. What's up?
The side effects of taking too many NSAIDs would be having like a GI ulcer, but you're not going to have any. It's not going to alleviate the problem.
So some of the claims out there in the general media, and I think it's not necessarily by people that are in positions of clinical practice, is that they were causing the virus to go into overdrive and it was feeding the virus. That's all nonsense. Viruses don't feed on NSAIDs. It's a compound that relieves prolactin, like reduces prolactin levels, not prolactin, prostaglandins. It actually blocks the prostaglandin. It's an anti-inflammatory. So if you block prostaglandins, it blocks inflammation. Is it going to disappear like the Spanish flu? Spanish flu actually never disappeared. H1N1 in 1918 was the same H1N1 that was the swine flu epidemic in 2008. So unlike 2008, we had a lot better grasp of how to treat the flu. We have Tamiflu readily available. We do have a better idea of how to quarantine people. Spanish flu actually was H1N1, but one was more virulent than the other. There was a slight change between seasons. It started out as not so virulent and so aggressive, but then summer came, dried everything out, and then the next season in 1918 was when it really got terrible for most people. But there was also a world war going on at the time. A lot of people in the trenches died from the flu, and that actually increased the numbers and made it more deadly than what it could have been had it not been during that time. The difference between that and now is that it doesn't appear that this virus is temperature sensitive like the flu or other common cold viruses like rhinovirus, meaning that you're going to see people in, say, Florida, the southern hemisphere, or anywhere else that the temperatures are incredibly high are still going to be able to spread this from person to person, and that it means that the virus can live on plastics and metals for longer periods of time. Now, there is some conflicting research as to how long the coronavirus, particularly this strain, can last on plastics, metals, and other types of surfaces. Tends to be that plastics are the surfaces that this virus can live on the longest. If you look at data from other Coronaviridae family members, including SARS, some other common colds, different things along that nature. So a lot of people are expressing a little concern about eating out or getting takeout and the packaging. I'm not saying that, the guy behind the counter is not cooking your food and isn't sick because he's got to go to work. However, basically, you touching packaging, say you get an Amazon package, if you need something, you need whatever other type of items that you need. The box that you get You don't need to sanitize it. You can by all stretch of the imagination, but you don't necessarily need to live in fear that you're going to get something from, say, a box. What I do recommend is that if you are going out in public, again, wash your hands. If you touch doorknobs, do not lick doorknobs. Do not go up to people that you don't know and just have a conversation with. If you're in places that are in shelter-in-place regulations right now. I miss you too, Joe. So there's a lot of conflicting things about that. However, the main way of spread is through droplets. So if somebody's next to you and they tend to spit a little bit when they talk, avoid them. The other thing is if you cough into your sleeve. Don't do as I've done a couple times and touch your face. I lost my train of thought. Don't touch your face and then touch other people. Basically the virus needs something to carry it from person to person, including droplets, mucus, stuff like that. There is no evidence to suggest smearing, basically taking and licking your hand and wiping it on a surface, will actually cause the virus to live longer, will die out, dries out. Actually, it's a very philosophical debate. Viruses don't die. They're not even alive. But that's for a different day.
That's true.
You know what I mean? Yeah. But there are some antivirals right now that are being tested. Remdesivir, as my cousin handed me the notes on. Anything that blocks RNA polymerase could be potentially used in this particular viral vector, meaning that if it can block the replication of the viral genetic code, then yes, that could definitely be used. There's a lot of antiretrovirals that they're investigating, including HIV drugs. I know that they were looking at some, in the one study I saw was penciclovir, which is acyclovir or an anti-herpes virus medication. It showed very little efficacy in terms of virion particles being produced. So it's just many different avenues that need to be investigated. You probably won't see a vaccine on the market for another 18 months, maybe 17 months at this point, maybe a little bit quicker, depending upon the lifespan of this virus. So if it keeps going for 18 months, you'll definitely see a vaccine. But if it's something like similar to the Ebola, Zika, and SARS outbreaks of the past, you probably won't see a vaccine come to market in time to actually do anything to kill the pandemic or epidemic from previous times, but pandemic this time. Does cooking your food at a high temperature kill it? If it's temperature sensitive, then yes, but most likely it should kill, in parentheses. Okay. But yeah, just cook your food properly. Wash your hands before you cook. Wash your hands during your cooking. If you touch your face, like if a person has a cooks, yeah, it's fine. You should be. In most cases, you should be fine. It's not a disease that's going to go through your food like that. It's most likely if someone coughs on your food after they cooked it. Would definitely be the mode of transportation. But high temperatures such as like 200 plus degrees Fahrenheit, 300 degrees Fahrenheit should kill any virus, particularly in the Coronaviridae family. Okay, lots of conflicting research on incubation. Some saying 10, is that long before seeing symptoms?
Is it about incubation?
So basically the question is, 10-day incubation, is that long before seeing symptoms? Is it 10 days before you see symptoms, or is it sooner? What is roughly the length of time being seen on length of virus life on surfaces like plastics? Plastics, for other members of the Coronaviridae family, it's about 24 hours. It could go up to 3 days. However, other family members, not necessarily this particular coronavirus, but all of their biology is pretty much the same. SARS numbers are 24 hours for metal surfaces, maybe 24 hours to 3 days for plastics. However, that's at basic room temperature, 25 degrees Celsius, about 68 to 72 degrees Fahrenheit. If you have temperatures that are much higher than that, it'll probably be much shorter. For the incubation period, I think I have a note for that. Based on data from the first cases in Wuhan and investigations by the China CDC and local CDCs, including members of our CDC that are in China, working with them, the incubation period could generally be between 3 to 7 days and up to 2 weeks, as the longest time from infection to symptoms was 12.5 days. And the confidence interval is very high on that, between 9.2 and 18. This data show that this novel epidemic about every 7 days, whereas basic reproduction number, or R naught, is 2.2, blah, 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 blah. So basically, you can see symptoms as early as 3 days after infection and as long as up to 18 days based on the statistical analysis. But that is also, again, early on. That study was only looking at China's data, not necessarily Italy's data. So I will have to you if that has changed. But 10 days tends to be a good number for the incubation period. That's why a lot of people who do test positive for COVID-19 are told to quarantine for at least 14 days, because that is at the high end of that incubation period. And it tends to allow the person to become healthy and not shed viral particles at all. So, I don't know. Do you guys have any other ideas that I should talk about?
Do you have any, I can answer some questions about...
Do you want to talk? Yeah.
Should I introduce myself?
Okay, I don't know. Can everybody hear yet? Give me a thumbs up or a thumbs down. It's my cousin Anthony. You want to sit? Yeah, he's a frontline nurse. He's at University of Penn. He'll know a little bit more about the clinical sides of things. If you want to talk about maybe...
I guess I can talk about what we're seeing in the hospitals a little bit.
Yeah, give people, because I know some people are Debbie Downers or naysayers about, is this real?
Sure.
Can you speak to like how, like, not the number of cases, but can you give us an idea of how the peak is going, how it is that we're flattening the curve, and stuff like that? Like,
just as a disclaimer, these thoughts are my own and not affiliated with my hospital. Yep. But I do work at UPenn Hub, the main hospital there. In terms of what we're seeing at the hospital, we're definitely still not at the peak yet. So a couple weeks ago, I remember I can speak explicitly to the first COVID patient that we had, and it was kind of like, okay, we have a patient now. Like, what do we do? Like, what are the policies going to be in terms of like protection, treating the patient, protection for people treating the patient, so on and so forth. Every day we've seen an increase in patients, and the policies are kind of changing as that's happening. I think the hospital system itself is at a point now where we kind of have procedures in place that are going to stick around and pretty solid, but it took us, a couple of weeks to kind of get here. In terms of the actual peak, more and more patients are coming in every day. We went from dedicating 1 unit to COVID patients, 2 units, 1 critical care, which are like acutely sick people, these people that are on breathing machines, different medications to kind of keep them alive. Can
you speak to how many people you see not on the ICU, but maybe on the second tier of severity?
Sure. So we went from having the 1 unit critical care and 1 unit non-critical care to now we have a whole building dedicated to COVID patients. So this building 7 floors. So that includes 3 ICUs at this point, and then 4 regular floors. In terms of like the ratio, so we're not seeing a ton of people in the hospital. Actually, most of the people that are coming with COVID the studies are suggesting that if you have a patient that's presenting COVID-like symptoms and they're in respiratory distress, which is one of the main symptoms in the later stages, that early intubation is better for these patients. So intubation is when we go and put a breathing tube in and take over their respiratory drive. And a normal patient that comes into respiratory distress, you kind of want to put off intubation as long as you can and give the patient a chance, because you're taking over someone's breathing drive and there's some complications down the road associated with that. But like I said, the research is kind of showing that early intubation is better on for these patients. So what we're seeing is that people are actually listening, and some of the guidelines are, if you're feeling these symptoms and you're not in distress, stay home, self-quarantine. So we're actually seeing that HUP right now is operating at like 50 or 60% capacity of total beds, which is unheard of. On an average day, since COVID, we're operating at anywhere from 108 to like 112% capacity. So people are actually staying home. So what that reflects is those people that are staying home, if they came in, they may have got admitted, right? You come in you come in febrile with a fever, some of the symptoms, your labs are going to be off. We may be like, oh, this is a COVID positive patient. They're not acutely ill. Let's send them to a regular floor. We're not seeing those patients. So the numbers in the hospital are skewed. So we're definitely seeing a lot more critical patients. But I don't think that's because there's more people out there getting acutely ill. I think there's the number that's definitely larger is the people that have it and aren't acutely ill. They're just, again, staying home. My position at the hospital, I'm a critical care resource nurse. So I work every ICU. I work PACU, which is post-surgery, and then I work in the emergency department as well. I actually had emergency department shift today. And just kind of throw some numbers. I mean, I think we had like 4 or 5 people coming out positive, all kind of got intubated, got sent to the floor.
So I spoke a little earlier about testing. Do they have bedside testing now, quick tests, or are they still doing the PCR-based analysis?
So we're still doing swabs. We're doing, and don't quote me on the exact protocol, but so when you come into the ED, it's actually a triage tent out front. They fire rescue, or if you drive up, they bring you to a triage tent, and then there's people out there that are trained to determine, okay, you could potentially be COVID and you could, or you're not. And from that point, before you even get to the hospital, you kind of go through different pathways. They're doing, one of the things they're doing is, I think they're still doing it, is a rapid flu test. So, because if you're not testing positive for rapid flu, I guess that you're not testing positive for COVID. So that would be like a rapid bedside, but then they're still doing the send-offs. So what we're doing when those patients are awaiting those results to come back, which can take some time, unfortunately, you're treated as if you are a COVID patient. The protocols in the hospital for COVID rule out, which is a patient that's been tested and hasn't been confirmed at this point, are what we call droplet precautions. So the theory is that the virus spreads by like droplet particles. So you have to get it on the mucous membrane, which includes your eyes or your mouth. So the precautions for that are just the regular masks. We have some.
Yeah, we actually have.
Actually, I have my whole bag upstairs, but so this is like if it's just a rule out, you put on this mask, you wear glasses. We're supposed to wear glasses with edges because the particles could enter your eyes. Personally, like a lot of people wearing caps, and then you have to wear a gown as well. Now, once they become intubated, once that breathing tube goes down to a COVID patient, the virus then becomes aerosolized, as they say. And then you have to go to a more strict mask. That's when you get the N95s. And if you aren't familiar, it's like, I have one upstairs, but I don't want to waste the time to go get it. It's like the mask you would see on maybe like a construction worker, or like they're kind of those are more like an airtight mask. These masks are like anyone can just throw it on. The N95 masks actually require yearly fitting tests to see, because there's 2 different sizes, and like someone like me, I got fitted for a mask, but you're not supposed to have a beard. So if you have one, then you get put in what's called a papper, which is like this spaceship looking thing that goes over your head, and like your whole head's encased, and it's hooked up to a battery, and there's like negative pressure. coming out of it. I don't know. It's pretty sweet, actually. Like, pretty cool walking around that thing. But again, like, supplies are low. So, like, we're reusing these masks. We're reusing the 95 masks. We're reusing the pappers. Just wiping a lot of stuff down. The pappers are somewhat reusable, but some of the parts probably shouldn't be. But it's kind of the nature right now. Now, if you're on the floor and once you're tested positive for COVID, again, you go to that more restrictive mask, but that's in the ED. If you're on the floor, it's papping. No matter, like, they don't even let you use a restrictive mask. So that's kind of what we're seeing in the hospital.
The same protocols. Okay.
In terms of the peak is coming. It's not here.
Yeah. So to put it in perspective, if you look at the way that New York was behind Italy, we're about, I would say, a week and a half behind New York City in Philadelphia. We're doing this out of Philadelphia. Cuomo and the New York health officials said that the peak death rate will not be for another 2 to 3 weeks. Peak infection rate could be anywhere between now a week to 21 days. But if you're looking at some of the curves, if you look at Italy, Spain, and the United Kingdom, you're looking at that the peak went up rapidly, and now it's kind of leveled off. The way that kind of flat is that you're going to see that curve, that plateau go for a little bit longer than you normally would if you had a quick peak and down. We're also not seeing the direct measures, say, that you would see in Italy with a lockdown. They're actually using police to enforce. In the United States, particularly, I know I can speak to Philadelphia. People are still outside. People are still congregating. People are still getting together. School River Trail is completely overrun with people. So if you can, stay home. But we're probably going to see that curve go on for some time. And then the critical people that Anthony are talking about, the people that are on ventilators, they're going to be there for a few days before they get to that, point where they die. Yeah, it could be quick. It could be quick and acute onset. But you're most likely going to see... Okay, and then you're gonna have the delay. So what you're seeing now is the infections that happened 2 weeks ago because of that incubation period. So people are going to get sick, going to show symptoms, go to the hospital. 4 more days, you're going to see people get more severe symptoms. This is on average, I'm assuming.
Yeah,
Yep. And then you're going to have people that go into that severe stage where they need the ventilator, and then they're going to have that death rate. So all the numbers that you see in terms of the deaths are going to go. That's fine. You're going to see lag behind the infection rate.
For sure.
So you're going to see a quick spike in infections, and that may level off. But deaths still may be increasing for 10 days after that. Yep. So that's the reason why you see the restrictions being extended, that we see the stay-in-place orders up until at least April 30th.
Right.
So
yeah, like,
there was a question.
We'll get to the questions in just a second, but just to kind of off what Dave was saying, if you look at the graphs, you're looking at 2 different graphs: infection, death, and you can superimpose them on each other. And like you said, one's going to lag. What people have to realize is that this virus is causing symptoms that we see in the hospital, right? It's causing what we call ARDS. You just go into acute respiratory, your lungs shut down. We see this in other infections, right? Mm-hmm. This isn't these people that get better, these critically ill people, this is a long journey for them. Mm-hmm. So the people that are going to get better, I mean, you're talking weeks to months on a ventilator. Mm-hmm. Some people end up getting trace, could be 6 months down the road if they recover. Some people don't recover. Like this we see all the, like, unfortunately, this is medicine in a critical care setting. It's just how many people are coming in and how quickly it's happening. I don't think Philly's, obviously New York is bad because of the population, like, and kind of how New York's set up, like, yeah, the density of the people. I mean, you take any block in Philly and you put that in New York, and you probably have 2 or 3 times as many people in that same amount of area. I think Philly won't be bad, and I think, so people aren't social distancing like they should be, but there is, you can clearly tell the difference. I mean, everyone can. And then
now we live in the same house, so it doesn't
matter. It's too late. But to answer some of these questions, the first one is, how much more likely is a person with 4 underlying conditions to get it, and why does it turn deadly on people with you?
So it's not that you're more likely to get it unless you're immunocompromised. If you have some type of immune deficiency, then yes, you're more likely to get it. But what they mean with people with underlying conditions is that the potential risk of severity of the disease is much higher.
Right.
If you have,
and it's dependent on what your underlying conditions are. It's attacking some conditions disproportionately, cardiac history,
diabetes,
Pulmonary.
and then obviously pulmonary.
Yeah. But it doesn't necessarily mean that you're more at risk of contracting the virus, unless, like I said, you have some type of autoimmune disease, or if you're taking some type of medication, what is it?
I mean, if you're on
Humira,
anything
Enbrel.
Anything that suppresses your system, which is common with most. Like if you have an immunosuppressive disease, I'm sure they at some point you've been taught like you can get sick more frequently than most people from anything, the flu, a regular pneumonia. It's just this is a drug.
We're basically staying at home for
steroids.
people like that, you know, people who have had chemo, radiation, who have compromised immune systems. That's why you're at the stay at home. You and me can get the virus. There's a lot of cases of people under 44. I think it's like 40% of the people that get it are under 44 and still go to the hospital with the severe symptoms. But it's more that you could be a potential carrier to people that are older, more sick. Different things along that.
So the underlying condition kind of in a hospital setting, we see people with underlying conditions get affected more severely by a host of infections. This kind of spotlight. Your body's not able. Let's answer Christian's question real quick. Christian asked about, is the hospital running any like antiviral tests on these patients, or what drugs are they using? So to speak to the drug we talked about earlier, in your school, and I'm terrible with drug names, but it was remdesivir. I should probably learn that before I have a test on it. But so that drug was produced by this company Gilead. I have seen that used. The issue with that drug is that it's still considered experimental, which is fine at certain hospitals, Penn being one of them. We give experimental drugs all the time with proper consent and guidelines. The issue is they're not giving that drug to people that are there's limitations onto who they'll give that drug to, because you want, not that they want the numbers to look good, but someone that's super sick that might be on a ton of medication keeping their blood pressure up and like a ton of life-saving measures, those people have a low percentage of survival to begin with. You give this in this type of drug, a lot of it based on underlying conditions. So you give that person this type of drug, the drug might help with the virus, but based on their underlying conditions, they're not going to do well. It sounds like
plus, you know, those the other drugs that they're taking could also interfere with the metabolism of whatever drug, you know, that they're trying to test. Like hydroxychloroquine was one of the ones that we talked about earlier. This remdesivir, other antiretrovirals, they may be metabolized and actually increase toxicity for the patient that may be detrimental to the patient in the long run. So it might actually cause more damage than good. Also, only certain locations, I'm not particularly sure about Penn, but will be with doing drug A, drug B, drug C. Another location might be Z, Y, X, you know, different things of that nature. So it's still all over the place in terms of what they're actually using. I think hydroxychloroquine is one of the ones that is most spread out because of the initial data in vitro showing that it can actually block viral absorption. But again, there's still months and months and months to go. And, you know, it's pretty much shooting in the dark right now to see what drugs we have on the market that can actually be like retrofitted to use for this particular viral infection.
What are they doing at? I'm not sure.
What are they doing at?
Anything like based off different precautions than the normal?
No, just doing normal drop precautions, and we're limiting only having parents be the only caregivers that are allowed in.
All right. So kind of the same precautions, limiting visitors, which every hospital at this point is doing. You can't get into a hospital unless your family members.
Possibility, we have 14 days that we're keeping them on, just to rule out.
Okay. They rule out period.
Just because you don't know, even if it's negative, does that necessarily mean from that exposure?
Right. pretty much the same kind of across the board.
Yeah.
Really, I mean, having asthma and autoimmune and meds, I would definitely be a lot more careful.
The asthma portion of it, though, the only thing that I would say is that you're not necessarily going to have much worse off conditions from the virus. However, some of the drugs that you use, such as adrenergic receptor agonists and glucocorticoids that you would use to treat asthma, can actually reduce the immune system of your respiratory tract, so they can actually give you a little bit of a leg down, so to speak, in terms of fighting off the virus. But asthma in and of itself shouldn't be too bad of an underlying condition. The autoimmune and meds, it depends. It really does. But that's only my take as a biologist, not necessarily as a clinician. So take that with a grain of salt. Big grain of salt.
Yeah. Just be careful.
Yeah.
Asthma is, a lot of things can kind of, as you know, precipitate asthma attacks.
Yeah.
I mean, again, we've talked about the differences with this virus compared to the flu and stuff, but in the end of the day, they all kind of fall into a broad category. And some of the various underlying conditions people have, the precautions you take with anything under the categories, you want to kind of apply the same caution, and if not more. And
just to kind of beat back one of the other myths that people are throwing out there, including Trump, is that it is not the flu. It is definitely not the flu. Even if you look at the more conservative numbers of the death rate, looking at infections versus death rate now may not be the best idea in terms of like what your percentage is. But if you look at Italy and Spain's numbers, they're averaging around 7 to 10%, closer to 7% of a death fatality rate. That is about, it's 0.5, goes into, it's about what? 10 times, I think, less, it's 10 times more deadly than the normal flu. So the normal flu, you have a death rate of what? Yeah, coronavirus in general. The flu definitely kills at maybe like 0.0, or is it?
Yeah, add to that, yeah. So I think the flu is down to like 0.4%.
No, the pneumonia vaccine you would have gotten would have been for
pneumonia. That's not going to help.
That's a bacterial infection.
If there were vaccines around, the world would be going nuts for it. So this is my personal view in terms of comparing death rates of this to the flu. This is one thing I have an issue with personally. Is that one? So the flu we have a vaccine for. So I work in healthcare and have to get the vaccine. Tons of people get the vaccine. I don't want to get into whether vaccine's appropriate or not. You can have your own views, but the thing is, there are people who are vaccinated against the flu. So that right there, that vaccination alone drives down to what we could say would be the true death rate of a flu. If there was no vaccine, if it popped up, if it was novel like this coronavirus. The second thing is this social distancing and telling people to stay home and not testing people. So there's a bunch of people that are at home right now, not feeling well, probably have COVID-19 and haven't been tested. So if you say, okay, 100,000 people got COVID-19 and 10,000 people, or 10,000 people died, right? Like this, let's use arbitrary numbers. You can say, oh, the death rate is 10%. But there could be another 50,000 people at home that haven't gotten tested. So that death rate is actually a little bit lower. So those are just my views on like where it's so, it's like, well, we're not testing. And also, people vaccinated against the flu, people that are highly at risk are definitely vaccinated against 65 or older. You come to the hospital at 60 through flu season, it's required by law for us to ask you if you've had the flu vaccine and if you want the flu vaccine.
Plus, on top of that, the flu numbers in terms of fatality rate, our system's built to handle the flu influx, that flu season. So generally speaking, we should have enough ventilators, enough ICU people, enough precautionary measures, vaccines, Tamiflu, different medications and stuff like that, that will also naturally suppress the fatality rate. Coronavirus, though, right now, if you increase the number of patients being seen, as you see in Italy, you're going to have that spike where you're going to have 100,000 people come in. I think it's 80,000. But you're going to have an unnaturally high fatality rate because you have
of
so many
resources.
people. Yes, your lack of resources. So then you have to triage. Oh, you're 60, you're 65, you're 70,
Yep.
but we have 10 other people that are 45 that also need the ventilators. In Italy, they had to choose whether they're going to give it to the younger people that are positive and that are in severe condition versus the older.
Yep.
So that's, again, why we're seeing the social distancing as the means to kind of protect against unnecessary deaths or the increase in fatality rate in that regard. But then if you look at it again, we're still at the tail end of flu season, are we not?
Yeah. We are.
We're still coming
Absolutely.
down off of it, but you still have that chunk of people that need ventilators, that need the infrastructure in place that we have, that, now coronavirus on top of that, there's only so much, water that you can fit into a cup before it overflows.
Right.
So that's basically what the overflow is.
Exactly. The flu at this point is mapped out and predictable, and to the point where hospitals know during flu season, like staffing is accounted for. There's more money in the budget to have more staff. Like it's more resources. We know this is how many people are going to come in. There's a range. We know how many people are going to be sick. We can take care of them. Coronavirus, we don't know. The system is starting to be. Some places it's busting out of it past capacity. Other places starting to get there, and people are going to unfortunately not get the care they need because of a lack of resources, and that's going to, like you said, not inflate, but cause it to be a higher mortality rate, unfortunately.
CO is a problem.
It falls under
it's
underlying pulmonary issue where it would be of concern.
Yeah, I would.
Stop smoking.
Yeah, keep it simple. Yeah, if you have any type of underlying condition, smokers, people like that.
If you're still smoking in 2020, no offense, you can yell at me later, but you need to stop.
Smoking's bad. Pretty much covers on everything.
Right?
Rosie, do you have any other things you think we caught?
No, we're good.
Okay. All right, everybody, wash your hands. Don't touch your face. Keep socially distant. If you have questions,
This dog is bad. This shit.
yeah, this dog's gonna eat everybody. Obviously, listen to your primary care physician. You are having symptoms, call ahead to whatever doctor that you're going to go to, including the emergency room. They do have phone numbers.
Please call.
Just call, and they'll guide you as to how to proceed. Don't use anything in this. What would you say to Jair? Yeah, he's an idiot. It's all a hoax. It's all bad. Anybody who thinks this is a hoax, anybody that thinks that this is a hoax, I will personally pay you to go into New York City, into the ICU, without any gear, without anything, and take video and spread it around.
I can tell you it's not a hoax.
It's
also, just personally, these are my thoughts, not my hospital's thoughts. So don't go telling someone Penn said this or that, because I don't want to get fired. No, I'm just kidding. But just have to do it there.
Yep.
Who else is on there?
All right,
Those are
buddy.
great.
Everybody, be safe and keep distant. Just keep distant. Listen to everything.